let me introduce Birte. So thank you very much, Birte, for having accepted this invitation. Sorry, I have to click on people arriving. So a few words about Birte. So she obtained her PhD in 2013. Am I right? Yeah.
14 officially, but 13 I submitted.
Okay. And it was at the International Max Planck Research School for Computational Biology. And then you went to Reykjavik in Iceland at Decode Genetics for 3 years. And then you went back to Berlin, where you were already a junior research group leader at the Institute of Health. And now you are in Regensburg. I guess I hope I pronounce it well. Since the COVID, say, since 2020, where you had the algorithmic bioinformatics research group. It's quite impressive CV. And so you're interested, among other things, in studying the human genome structural variants in a biomedical context. And this is what you are going to talk to us today. It's how to detect and genotype structural variants using short reads, if I'm right, and a linear reference genome, but also on some graphs, representation of population genomes. And once again, I thank you, and the floor is yours.
Yeah, thank you, Pierre. Exactly. I'm going to mostly speak about linear reference genome or short reads aligned to a linear reference genome, but added some slides about considerations for graph genomes as well, given this audience here. Yeah, so the main focus, as Pierre said, is really the development of methods for studying structure variants in my research group. And I think structure variants are very interesting, not just because they contribute significantly to human genetic diversity, as maybe even some people here published a few years ago, but also because they contribute to the risk of many complex diseases and can cause rare diseases as well. And here I'm showing one example from my own research, a GWAS for myocardial infarction, where we identified a 766 base pair variant, decreasing the risk for myocardial infarction, and an example from a human geneticist in Berlin, Stefan Mundlos, who's studying limb malformations at Charité and the Max Planck Institute, and he has described several deletion, inversion, and duplication variants that can cause these limb malformation phenotypes. So also in the context of disease, structure variants are super important and interesting. But let's start simple. What are structure variants? So by definition, usually people nowadays say a variant is a structure variant when it affects at least 50 base pairs of sequence. There have been reports of more than 10,000 or 10,000s of structure variants per human genome. This is the current estimate, and I'm sure it will increase. Maybe not to hundreds of thousands, but at least a few more tens of thousands, I would guess in the next years. Each individual structure variant is more likely to be relevant for disease than an individual SNV or indel, just because it's so much bigger. It affects more sequence, so it's more likely to hit something important. And structure variants are also more difficult to detect in genotypes than small variants because they often occur in regions that have repeats. It's due to their formation mechanisms, and also they are more diverse. So they include deletions, insertions, duplications, inversions, translocations, and then also the complex structure variants. So complex structure variants are always somehow combinations of the other types of structure variants. For example, a deletion next to an inversion, and people often just list them as complex structure variants. But I'm going to talk a little bit more about complex structure variants. So I've brought a few examples of complex structure variants here. So this is an example of an inversion combined with a deletion, a duplication with an inversion, and then you can make this arbitrarily complex actually and combine several of such events together, and it would still be something that we call a complex structure variance. People, when I talk about complex structure variants, people often ask me how many are there actually in the human genome. There are not many per genome, but I think it's still an interesting class of variants. In NOMAD-SV, for example, and this was solely based on short read sequencing data, Collins and others discovered more than 5,000 complex variants out of the 433,000 SVs they described. So it's still a considerable number if you look at many genomes, and I think we should include them in when we study the causes of diseases. And they are still an important class of variants. So if we want to study them, then because they are so heterogeneous in both size and complexity, so also the sizes can differ from just a few base pairs to many thousands of base pairs. Because of this, we need very general analysis approaches, and it turns out that we have to generalize everything in the approaches for detecting and genotyping complex structure variants, including, and this is what you do first when you start studying them, the description of structure of complex variants. So this is very descriptive, saying, okay, here's an inversion and next to a deletion, but it's actually hard to use this in a very generalized approach. So the first step, what we did is we said, okay, we specify complex structure variants in terms of their breakpoints and novel junctions. So breakpoints are the positions on the reference where you would break up the genome and then put these pieces of sequence together, creating novel junctions of the pieces of sequence that you broke up on the reference genome on the alternate allele to obtain the variant allele. So breakpoints are on the reference allele and novel junctions are on the variant allele. And each breakpoint or novel junction connects two pieces of sequence. If you look very closely in this picture here, you will notice that the novel junction 1 and novel junction 3 in this example, and also two novel junctions down here, are actually not novel when you look at the reference allele, but they are actually piecing together the exact same pieces of sequence as on the reference allele. We still keep such junctions in our set of novel junctions for the description of the variant because we think it's useful to have them, although they are not really novel junctions. Yeah, as Pierre also said, we're using short read sequencing data for studying complex structure variants, even though a lot speaks for using long read data, especially the fact that the majority of SVs has remained undetected in previous studies using short read data. So when using short read data, the 1000 Genomes Project only discovered like about 3,400 SVs per genome, and NOMAD-SV and also the recent UK Biobank data analysis by Decode found less than 8,000 SVs per genome. When you go to long read sequencing data, then you find more than 22,000 structure variants per genome. So this is really in favor of using long read sequencing data for studying structure variants, and especially the complex structure variants. But there are also other things you need to consider when studying such variants, and one point is that the abundance and availability of short read data is still so much better than for long read data, and that's because the price is still higher for long read sequencing than for short read sequencing. And my guess is that this will stay like this for some time, that we will have much more short read sequencing data because people invested in this, and we should try to get the most out of these data sets that have cost a lot in the past and are very valuable data sets. So we should do our best in extracting the most information from this data. Okay, back to structure variants. There are two problems you can address when studying structural variants. One is the detection problem and one the genotyping problem. And sometimes people forget that these are two different problems. I've been mostly looking at the detection problem in the past, developing population scale approaches for identifying structural variants in many genomes simultaneously. So this problem asks where in the genome are what kind of variants. And I always thought this is the more exciting problem because you discover something new. But after having prepared several call sets for downstream analysis, I realized how important the genotyping problem actually is for the downstream tasks. So you need accurate genotypes in order to do something with the call sets, especially if you have called variants in a large number of genomes and are not just studying a single genome. Then your genotyping needs to be accurate. So this is the question: how many chromosomal copies of a given genome carry a known variant, a variant that you know that is occurring in some genome in the population. So you know the variant already and just want to know what is the genotype of a given genome. And this is going to be the focus of my talk today. We just 3 days ago uploaded a preprint on bioRxiv describing this method and submitted it. So hopefully this will be published soon, but if you're interested in details, you can already check it out on bioRxiv. Yes, so when you start working on a problem such as genotyping, you always have to, of course, look into the literature and see what kind of approaches other people have used. And I now categorize them into 4 types of approaches. The first one type is the classic approaches that use the reads aligned to a linear reference genome. And one such program that does that is, for example, SVtyper, which is not published as a standalone tool in Nature Methods, but it's just part of a pipeline, and somewhere deep in the methods, the SVtyper tool is described, and people have been using SVtyper a lot in many papers. So this is why I'm mentioning it here, and also in many other tools, this classic approach has been implemented, including one of my own tools. So you use reads aligned to a linear reference genome and then somehow classify them as variant and reference supporting based on read pair signatures. And for deletions, it would, for example, be the signature where the reads align further apart from each other, the reads in the same pair, such that you can infer that there was some sequence deleted. Or the orientation for inversion variants, for example, as a signature. This is based on linear reference genome alignments. Then there are already a few tools for pangenome alignment. One is, again, deep in the method section of VGGRAPH and a VG paper by Hickey. It's described where you use reads aligned to a pangenome graph, and then you look at read pairs that align along the graph edges, and those are informative for your variant, and actually based on coverage along the different paths through the graph, a genotype is output. A combination of these two things is the graph realignment approach, where you use reads aligned to a linear reference genome at first, but you extract the reads from a locus where you know that, or where you want to genotype a variant, and then you create the graph of this variant, just a local subgraph, and align your reads to the subgraph, and then use a similar approach as in the pangenome graph alignment approaches. This was actually earlier than the pangenome approaches, for example, implemented in Paragraph, and also GraphTyper 2. And you obtain then the genotypes and genotype likelihoods from the graph alignments. And then the last class I introduced, or I decided to put here on the slide, is the K-mer-based approaches, although you could also put these approaches in other classes. But I decided to have a K-mer-based approach class for base type and PANGEA because they don't use an alignment at all, but just a FASTQ reads and then count K-mers or per haplotype, and then use haplotype information and the K-mers on the haplotypes in order to infer genotypes. In terms of methods, I think these are the most innovative and interesting methods, the K-mer-based approaches, although this also has some drawbacks, and actually all of them, if you look closely at them, have some sort of drawback. For the classic approaches, most of them only genotype a single breakpoint or novel junction, but not the entire variant. For the graph realignment approaches, this is actually true as well, but some people, such as the paragraph authors, have realized this and added some heuristics for afterwards combining the calls from the different junctions from the breakpoints into one call. But this is extremely heuristic, and when you have conflicting calls from two junctions of the same variant, it's really hard to decide what to report to the user of the tool as a genotype. If you do that afterwards. The pangenome alignment methods, as far as I understand, and you may correct me if I'm wrong, do not compute likelihoods so far. At least these that I looked at don't, which is something that should be definitely added if this should be used in downstream analyses. And the bigger drawback is that it requires the presence of the variants in the graph before the expensive alignment. So if someone comes to you and says, I have found this new variant here, can you genotype it in our 300 genomes that we have on our server and tell me what the frequency of it is? And you have alignments to a pangenome graph, but this variant is not in the pangenome graph, then this method simply does not work. Then you have to go back to these other methods again. And a similar problem occurs for the K-mer-based approaches because you don't rely on an alignment. Then these genotypers are extremely computationally expensive. I mean, you save the alignment, but usually you compute an alignment for other analyses as well. And then genotyping with these approaches is expensive. And another drawback of these methods is also that they are sensitive to small breakpoint inaccuracies. So if you have just an extra base somewhere inserted at the breakpoint that you did not specify in your variant description for whatever reason, then the K-mer-based approaches will break there at this point. So there's room for improvement, is what we decided, especially for complex structure variants, because many of the tools we also looked into are not able to genotype complex structure variants. And I'm going to start now simple, very simple, especially after I saw that here are some approaches that do not compute likelihoods at all. I just added some slides from my lecture for the master's students into this slide deck. And so this is the basic genotyping idea. So you have, or problem, you have as input aligned read data for a particular individual at a particular site, so at a variant site, a known variant. And you want to output likelihoods of the genotypes given this data. And the genotypes can be the homozygous reference genotype, no copies of the variant allele, heterozygous genotype, one copy of the variant allele, and then the homozygous variant genotype with two copies of the variant allele. And the most common approach, I would say, most widely used approach is a maximum likelihood Bayesian classification approach for computing genotypes. So you calculate likelihoods using Bayes formula for each of the three genotypes, and then you report the genotype with the highest likelihood as the genotype, but you also have the likelihoods for the other genotypes, which is important if you want to add some measures of confidence of your call. In Bayes' formula, you can actually ignore the probability of your data because it's going to be the same for all 3 genotypes, and then it cancels out. So we can ignore that, and then we are left just with the prior probability and this conditional likelihood here, which we split up in a product over all reads, assuming independence of the reads. And then this most interesting part here, and now I can't forward my slides. Here we go, is the likelihood of observing a read pair R when the underlying genotype is GI. So this is the likelihood we need to compute somehow we need to define in order to implement a genotyper. And the rest is more or less standard and used many times before. Yeah, so a very simple genotyper, for example, for a structural variance could be one that uses constant priors, 1/3 for all 3 genotypes, and then classifies read pairs in those that are variant supporting and reference supporting somehow. Yeah, there are lots of things you can consider when classifying the read pairs you observed, and then a reference supporting read pair for the reference genotype will have probability close to 1, and for the homozygous variant genotype will have a probability close to 0 and 0.5 for the heterozygous genotype, and vice the opposite for the variant supporting read pairs. Something extremely similar is implemented in SVtyper and also in SVGD that Pierre will know, only that they correct for the number of breakpoints in some way per variant. But it's actually quite similar to this approach already. If you want to go a little bit further than this, for deletion variants, for example, I'm showing you what we implemented in the tool POPDA. We used here an empirical insert size distribution, and instead of using 1 for variant supporting and epsilon for 1 and epsilon, or 1 minus epsilon and epsilon for the homozygous genotypes, we now use values from this empirical insert size distribution. Just the normal, the distribution that is observed in the data for the reference genotype, the distribution shifted by the deletion length for the homozygous variant genotype, and then the average of the 2 for the heterozygous genotype. This is something that works for deletions, but for example, for inversions, it doesn't help because variant supporting genotypes are, you can identify those best by the orientation. So for complex structure variants, we have to combine such ideas somehow together and have something general, and we implemented this now in a tool that we call Generalized Genotyper, GGtyper in short. And it uses exactly this approach of computing the likelihood as a product over all read pairs. And the idea is to model the probability distribution of read pairs that we can expect at the variant locus when the underlying genotype is GI. And then once we have this probability distribution, we can simply look up the read pair probabilities in this pre-computed table. So once you have a probability distribution which is specific to a variant, it's going to be easy to look up the read pairs you have actually observed in your data set. But the tricky part is defining this probability distribution. And I'm going to show you how we do this over the following few slides. So here's an example. Again, this deletion inversion variant. Just a very small toy example this time with some sequence. Of course, in real structure variants, these pieces of sequence will be longer, and also in the following slides I will show some reads of length 3. This is of course a toy example. And then once we have this variant, we can list the sequences for the 3 genotypes, and so we would have twice the reference allele for the homozygous reference genome genotype, and so on and so forth. And why I'm listing this in so much detail here is because this picture nicely shows that the total number of read pairs we will expect from these 3 different genotypes differs because the variant allele is shorter than the reference allele. So the total number of read pairs per genotype is going to be different, and the total number of read pairs is something that we are going to use to normalize some read counts to obtain the probability distribution. What you also can see is that you have different numbers of breakpoints here. So 3 breakpoints in the reference allele and only 2 novel junctions on the variant allele. So when you just genotype a single breakpoint novel junction pair, it's hard to, at a time, it's hard to tell how you would combine the calls from the different novel junction breakpoint pairs to into one call. So our approach actually takes everything into account together, at the same time. Yes, so I said our idea is to model the probability distribution of read pairs at the variant locus. And for that, we enumerate and categorize all possible read pairs from the reference allele around the breakpoints and variant allele around all novel junctions, which gives us allele-specific profiles, one profile for the reference allele and 1 profile for the alternate allele. Then we weight these read pairs using the insert size distribution. I'll show that in a bit. And once we have the allele-specific profiles, we always mix 2 allele profiles into 1 genotype-specific profiles and normalizes it. And this is actually then our probability distribution for the genotype. So let's have a look how we enumerate and categorize read pairs. Let's look at this example variant again, and we will enumerate read pairs both for the variant allele and for the reference allele. And I'm listing here just all read pairs with reads of length 3 and an insert size of 8. But we would actually repeat this for all possible insert sizes. So for all positions and all possible insert sizes, we enumerate read pairs from the variant allele and the reference allele. And then we can categorize those read pairs. And for the reference allele, we have here these 4 categories because we have 3 breakpoints and then 1 extra category for read pairs that are just normal read pairs somewhere in between the breakpoints or before and after. We include some piece of sequence before and after the variant as well. You can see I only labeled read pairs here that actually where 1 read in the pair overlaps the breakpoint. We could also include read pairs that have the breakpoint in between. We discussed this like for several hours, I guess, already, whether it is an advantage to do so or not. And we decided for now not to include them because we actually also have another category, and this will only be visible when you look at larger insert sizes than 8, where 1 read in the pair overlaps the first breakpoint and another one the second breakpoint, or you could also have the second and third breakpoint and so on. And these would all be separate categories. And if you have then a read pair that overlaps or where 1 read is before this first breakpoint and after the second breakpoint, and in between you have a deletion, then it's not going to be clear whether it is a read pair supporting the reference or the variant allele, but the difference will only be in insert size. And if the deletion is short, it's going to be difficult to distinguish. And we, for now, we have decided to include only categories that nicely distinguish the different alleles we have in of the variant. And this is true for reads that really overlap the breakpoint on the reference allele. So we can say for sure that this read pair originates from the reference allele. For the variant allele, we do the same, only that we include this time these read pairs that have the novel junction in between. And that's because we then, since we are using a linear reference genome, the observed reads will be mapped to the reference genome. So we actually, once we have enumerated the reads from the variant allele, we have to map them to the reference allele, because that's what we will compare to the observed read pairs. They are only aligned to the reference genome. So it's simple to map the reads before and after the variant. They will just map the same. Also inside the inversion, they will just be flipped, but otherwise align the same. The interesting parts are going to be the read pairs overlapping the novel junction. So for the left novel junction here, it will be reads in forward orientation, and for the right novel junction, it will be read pairs in reverse orientation. And these are actually the read pairs we will observe from the variant allele when aligned to the reference allele, assuming alignment is perfect, which is of course an assumption only. Yeah, and here we have then the categories of read pairs that are split at one of the novel junctions. So where the novel junction is actually the read pairs overlapping the novel junction on the variant allele, so it's going to be split, I should say, due to the novel junction or at a breakpoint. And then then we have the read pairs in forward and reverse orientation, which are also nice evidence for the variant. And then in the end, the gray read pairs that are the boring ones that just look like normally aligned read pairs without any indication of a variant. So to summarize this, we will have like 9 categories for this example variant, which we can classify our read pairs. The read pairs that we enumerated from the variant and the reference allele, and I remind you that we repeat this for all relevant insert sizes. So it will be lots of read pairs that we have enumerated and then categorized. So now read pairs that have different insert sizes are not equally likely to occur in the in a sequence genome, because in the sequencing process there's a fragment size selection process that selects read pairs following some sort of length distribution, or not read pairs but DNA fragments that follow some sort of length distribution, insert size distribution I've shown you before. And this is what we use to then weight our enumerated read pairs for the allele specific and read pair profiles. And then after weighting them, we mix the read pairs into genotype-specific profiles that we normalize into probability distribution. So this is illustrated here. Imagine this is the reference and variant allele where we enumerate all breakpoints, all read pairs of all insert sizes. Here we have the insert size distribution, which is library specific, so it's going to be different in every sequencing experiment, and we can measure it using the some sampling from the entire genome in the BAM file. And then we can multiply essentially the read counts per category per insert size that we've enumerated before with this library-specific insert size distribution. And in the implementation, it's really just a matrix multiplication that we perform here. And then we obtain an allele-specific profile for the reference allele and one for the alternate allele, the variant allele, which is essentially a set of insert size distributions per read pair category. So once we have these allele-specific profiles, we can then combine them into genotype-specific profiles. It's essentially a very simple linear combination of these allele-specific profiles. For the reference genotype, we just use something close to 1 times the reference profile and something close to 0 times the variant profile, the opposite for the homozygous variant genotype, and for the heterozygous genotype, we just add the 2 profiles in equal parts and then normalize this genotype specific profile to obtain the probability distribution, the probabilities we're interested in for calculating our genotype likelihoods. So this is the probabilities we obtain from our from mixing the allele profiles and normalization. And then once we have a data set from a genome, it's just a matter of looking up the read pair probabilities in this pre-computed table. So calculating likelihoods has quite a few advantages. And one advantage is that you actually can filter based on the confidence in your call, and one widely used standard measure of genotype confidence is the genotype quality, which is simply the ratio of the best genotype to the second best genotype, the likelihoods of the best to the second best genotype. While working on this, I actually went on maternity leave in between, and this is actually something that I now can recommend to everyone doing, because my postdoc afterwards came and said, well, this genotype quality, it actually doesn't take into account the posterior distribution of Q. So let's do some bootstrapping. They had already implemented it when I came back and calculated this genotype certainty, which is essentially just a fraction of bootstrap samples that agree with the genotype called GI. It's another measure of genotype confidence, just like the genotype quality, only that it takes into account the posterior distribution. And I will show you some results how the two metrics compare. And the second thing that happens, and this time it was my PhD student, he said, well, actually we have these probability distributions per genotype now. What about we just compare these two or 3 distributions to each other, and then we can already say whether it's going to be easy or difficult to genotype a given variant before actually looking at data, and we can estimate how much data we need to genotype a given variant, and so he implemented this as well while I was away. So it's not my contribution this time, but really my student's idea. So he computed the Kullback-Leibler divergence of the genotype profiles. So this is the relative entropy, which he had learned about in another course at university. And then the maximum of the inverse Kullback-Leibler divergence we now defined as the difficulty of genotyping a given variant V. And then we tested also whether this measure of difficulty actually predicts how well we genotype our variants. How difficult it is and how good it agrees with the precision and certainty we achieve. Yeah, so before I talk a little bit about graphs, at the end I'm going to quickly show you some results. We use simulated data, 200 genomes from the Polaris cohort, which is essentially 200 genomes or 150 genomes from the 1000 Genomes Project, including 50 trios, so 150 genomes from 1000 genomes plus 50 children of parent pairs in this cohort, and then some Icelandic genomes, including 3,440 trios for evaluation. On the real data sets, we used 20 handpicked common complex variants from different sources for now, and then compared on the Polaris and simulated data to other tools. For evaluation, we used on the simulated data recall and precision. Recall is just a fraction of genotype calls passing filters. So again, we filtered based on genotype quality or certainty, and also sometimes something else. And then the precision is the number of correct genotype calls divided by the number of genotypes calls passing the filters. If you disable filtering, then it's just the fraction of correct genotype calls of all genotype calls. On real data, we don't have any gold standard call sets for complex structural variants yet. At least I'm not aware of any. So we use the trio data, first of all, the Mendelian inheritance error rate, but this has issues, especially if you have, the parents have the same genotype. You can't compute the Mendelian inheritance error rate for many genotypes. And then the transmission rate is the rate of transmitting the variant allele from a single heterozygous parent. So when one parent is heterozygous, the other parent is homozygous, either reference or variant. And then the transmission from the heterozygous parent is expected in 50% of the cases. So you expect the transmission rate to be close to 50% if you have good genotyping. For unrelated genomes, there's another measure we use, the chi-squared test for Hardy-Weinberg equilibrium, which is a population genetic measure, or it makes lots of assumptions about the population, which are all not true. We know that, but it's a good approximation, and it works quite well in practice, at least if you have many genotype from the same population. So here you compute the allele frequency from the observed genotypes. Based on the allele frequency, you can then compute the expected genotype frequencies again, and then you can compare the observed genotype frequencies to the expected genotype frequencies. And I'm showing this in this plot here, where you have in red the expected genotype frequencies, and in green, blue, I don't know what this color is, the observed genotype frequencies. And this would be a nice agreement here for a variant that is well genotyped, and this would be something that fails the chi-square test because the expected and observed genotypes differ a lot. Okay, but let's have a first look at the simulated data. So here we could show that the genotype certainty, which is the red line here, is actually a better filtering criterion than the genotype quality, which is the blue line here. So you can achieve full recall if you just don't apply any filtering, and you can get higher precision if you apply higher, stricter thresholds for the quality or certainty. If you want to have perfect precision, you also need to filter based on average mapping quality of the repairs at the locus. So the alignment quality really matters here if you want to get full precision. Of course, you lose a few variants if you do so. And then this plot down here is showing the agreement of the calculated difficulty score for a variant with the precision and certainty. And except for the precision for one type of variant, there's a nice correlation between the difficulty and the genotype certainty and the precision. So we think this genotype difficulty is something that might be useful, and it's only possible because of the way we designed our method. So this is something we actually kind of like here about the approach. When going to the comparison to the other tools, we used base typer and paragraph. These were two methods which supported complex structural variants, although they have not been systematically tested on those variants, and they don't require a graph alignment. So we started out with linear reference genome alignments. And it seems that base typer outperforms GG typer in terms of precision, at least if you apply filters. No, this is without filters. to GG typer. But paragraph falls behind, especially on tandem duplications here and some complex types of variants that are shown here. And this is just zooming into the precision and recall on different coverages again, where you can see that base typer actually outperforms our tool on the perfect simulated data. And what I thought is interesting to see here is also that the complex variants, the solid line, are easier to genotype than the canonical variants. And I think it's because they have more breakpoints, more novel junctions. So there's more evidence for these variants. And this is why our confidence in genotyping is better and precision is better. It's a bit counterintuitive at first, but it makes perfect sense if you think about it a bit more. So on real data, this is when we saw this is when my PhD students started to panic a bit. Base typer is better than us. But when you go to real data, the picture changes quite a bit. So base typer, exactly what I described before. If you have small inaccuracies at breakpoints, it breaks down, and it actually didn't genotype a single heterozygous or homozygous variant call in several variants, and fails Hardy-Weinberg equilibrium for a few more variants, while paragraph and GG typer have very similar results and largely agree. We zoomed into one variant here, on these slides, I zoomed into one variant here where we have a close-by SNP that is in high LD with the variant, at least when we trust the genotypes of paragraph and GG typer. It looks here as if base typer is even better, has a smaller chi-squared value than GG typer and paragraph, but this is because it had genotypes many individuals as heterozygous, and if you do so, then it's easy to reach a high chi-squared value, and this is also why we took out these 4 variants where we have we have actually everything. Yeah. Oh, sorry. This is homozygous reference genotype, where we have only the homozygous reference genotype. So I think this nicely shows that on real data, the k-mer-based approaches can be very sensitive to the specification of the variant. And just for completeness, I'm showing the results for the Icelandic data here, where we have a Hardy-Weinberg equilibrium chi-squared value here. 3 variants are not in Hardy-Weinberg equilibrium. You can see that these are the variants also that fail on the trio data with Mendelian inheritance, error rate, and transmission rate. Although I think the Hardy-Weinberg is actually more informative here. Okay, just 2 minutes at the end. What about graphs now? So graphs actually seem to simplify this whole process. So when you take this example variant again and construct the genome graph of this variant, then each breakpoint in each novel junction is going to be an edge in the graph, and each allele is passed through the graph. And when you now go through the steps of our method, we can still enumerate all read pairs from the two alleles sequences for all insert sizes and all positions. And we can still assign them to categories, and we can even use almost the same categories as before. And what we could use all these categories here or just a smaller set of categories. It doesn't really matter. Let me go forward here. What we do not need to do when we have a graph genome alignment, we do not need to map the variant allele read pairs to the reference allele read pairs. And this is actually the most tricky part to implement in the tool. So we can skip this tricky part and then just pre-compute the genotype probabilities per category and insert size immediately from the enumerated read pairs of both alleles, and then categorize observed read pairs and look up their probabilities in our distributions and calculate genotype likelihood just as before. So actually, when you have a graph alignment, our method is applicable just as before, but it's actually a little simpler, and we simply just do not have had the time to implement it. Otherwise, we would have done that, and I would have been able to show you some results. But it seems rather straightforward and simple. We haven't tested it, so I can't guarantee anything yet, but this is what I observed when thinking about it. So yeah, in summary, we have, just to summarize what I've told you, we've developed a new genotyping approach for complex structure variants for short read sequencing data, given the description of the variants novel junctions. So we pre-compute these genotype profiles, probability distributions, by enumerating and weighting read pairs from all positions along the variant and reference allele for all possible insert sizes and categories. So this is the core of the method. So we get these profiles that we can then use also to compute the difficulty. And our approach outperforms paragraph on simulated data and base type on the Polaris data while achieving high accuracy on Icelandic data. So we think it's a useful tool, and it's going to be the tool that I use for genotyping structure variants in the future. By the way, it doesn't only work for complex variants, but because it's a generalization, it also works for canonical variants. Just a simple deletion can also be genotyped with this approach. It works just the same. Yeah, there are many future directions. We would like to include a larger set of real variants in the evaluation. For example, the NOMAD complex SVs. We have already implemented an extension for multi-allelic variants, so variants that have not just 2 but 3 or 4 alleles, and we would also like to test it on alignments up to the T2T genome and see if it improves the cause. Currently, we do not support variants containing non-reference sequence and also do not recognize unknown alleles. This is something we plan to add. And then support for long-read data and graph alignments as input is also been in discussion here. And we hope at some point we will have the time to implement this. One last comment at the end. One limitation of this approach that is alignment-based is that we will never be better than the alignment and the reference genome we're using, no matter whether it is a pan-genome reference or a linear reference genome. So there have been reports of using the T2T genome would simply improve the SV calling standard pipelines without changing anything in the pipeline. So moving to this better reference genome will probably already help. And then using graph genomes here, an example where some people have found twice as many as variants using pan-gene, though with a fancy approach for genotyping using on a graph, pan-genome graph reference. And with that, I'm at the end. I'm sorry that I took so long, but I hope we'll have at least a few questions. And also, if you know someone who's looking for a PhD position, we are currently hiring. Thank you.
Thank you very much, Bert. So I'll clap along, but I guess everyone is clapping. We don't have time for much questions, but we can take a few of them. So I don't know if someone has some question. There is at least one from George. Hi.
Hello, do you hear me?
Yeah.
Can you hear me? Yes. Okay. So thanks for your talk. It's really nice. And I have two questions. One is technical, and the other one is more about your opinion about genomes. So I will start with the technical one. When you mentioned the callback library divergence, the question is very simple. Would you prefer to have a metric instead of this divergence? I mean, is it useful for you to have the metric to maybe work in a vectorial space, or if you have a metric, you can also use manifolds to explore this I'm not sure if this is something of
I
interest.
think I didn't get the first part of your question. Could you repeat that? Or maybe Pierre, because I have trouble understanding what you say.
So when you mention the callback labor
Yes.
divergence,
Yes.
okay,
Yes.
Mike, this is not a metric, okay? That's
Yes.
my point.
Yes.
So
It's
if you change to the Wasser-Sein distance, you are in a metric space. So this opens many possibilities to explore things, right? So my question is this something important or is just part of the method and you don't need to change it?
yeah, actually, we haven't looked much into this difficulty score yet. We just saw that it nicely correlates, and there's probably much more you can do, but it was just an additional idea to add to the output of the tool, that it can compute this difficulty, and probably you can go further. The question is how whether people are going to pick it up and use it in practice, and this is what matters for me. If it's useful for practice, yes, but if nobody is interested in it, yeah, the question is whether we should spend a lot of time on it. But yes, I'm sure you can go much further there. Yeah, would be interesting to discuss.
Yes, and yeah, and also you can use manifold, but maybe this is not very well known. But manifold, you just need some elements for which you know a metric, and then you treat it. You can use many tools from topological data analysis that are being used in physics, but now you can use them for many things. I mean, manifolds are very general. You just need a metric, so maybe it's useful to think about this. And the other question, I just sent a link to a paper that was released 4 days ago, and the title made me think that it was actually for scaling pangenome graphs, but I took a look at the paper, and it's basically about how to obtain high-quality assemblies for human pangenomes. And this is a group from Singapore, and I asked them why they put scale pangenome construction, and the goal is to, in Singapore, they have a lot of ethnicities, so they need to create high-quality human pangenomes to then create the pangenome graph. The goal is to create the pangenome graph, but this paper is about high quality. So my question is, I guess that the high quality is because they really care about SNPs, but you care about the structural variants. So in terms of quality, is this important for you in this work, or you are okay with not having the best high quality assembly?
Yeah, in general, you can already call structure variants when it's not as high quality, but having high quality is always better than not having it. I would answer here. So especially the breakpoint inaccuracies, we are robust to it to some degree, but of course, if it's too inaccurate, all approaches will fail at some point. So it depends on
create
high enough quality, I would say.
And in your slide, you show breakpoint, and each nodal junction is an edge in the graph. You wrote in the last slide, right? Yes. But I think that, I mean, this is an assumption, or this is something that you want to see in the graph? Because I don't think that the heuristics, I mean, every tool to create a pangenome graph has an heuristic, and I don't think that they model this, but you could add it as a part of the model. So the question is, do you want to use the graph to discover them, or do you want to use the graph to create and to show this in the graph? So one thing is discovering the graph, or the other one is to, given that you already identify these breakpoint and nodal junctions, you would like to create a graph that shows you what you grow there.
Yeah, so I guess maybe I should, let me explain it here. This is, of course, when you create a graph from a variant description, you would create the graph like this. But when you have a pangenome graph already at hand and you identified a variant in there, you can just go ahead with that description of the variant. You don't need to have such a description. What you need to have for the approach to work, for the genotyping approach to work, is a path through the graph that corresponds to your alleles. And then also the points in the graph that distinguish the alleles from each other, and for us it was the edges here. So we use the edges as a basis for defining these categories in the approach. So this is what you need. in order to use the approach on a pangenome graph. But it's not necessary that you can map like the variant description like this from the linear representation to the graph representation. So we're actually kind of flexible here and can use whatever is in the pangenome graph. Although I have to remind you that using pangenome alignment only, so if you just rely on what is already in the graph, you can't genotype additional variants. So this is really a limitation there.
Yeah, because to identify it, you need to align the reads to the graph. But depending on the tool, for example, you use Graph Aligner, then you need that the seed is inside the node. So actually, the graph should be built such that it maximizes the potential for the tool to be used, and not the other way. So, for example, with the BG giraffe, they do not index the graph; they index the haplotypes, and then they use the graph.
Yeah, this is fine. This doesn't matter.
Different tools.
This doesn't really matter to this approach, as far as I can tell. Maybe you can teach me differently, but as far.
No, I don't know. I was asking. I just exploring this. But thank you. Yeah. Thanks.
Okay, thank you. Are there some other questions in the audience? I had one by Pio. Come on.
Hello, Birte. Just a quick question. If I understood correctly, when you were categorizing the different reads that you will read pairs that you will use, you were not using the reads that do not overlap the breakpoint, even if one of them is in the deletion. And my question is, why don't you use them if I understood correctly? Because they would also would support the fact that the deletion is not there, no? Yes,
you're right. And this is what we have been discussing for hours already, whether we should use them or not. And actually, I already suggested to a bachelor student to test different category specifications and the differences in the genotype calls in the end in her project. So we are not sure how much it matters and how much it affects the results. I'm sure it has some information, but then there's also some uncertainty for some of the read pairs that overlap a breakpoint and don't hit the breakpoint, whether they are from the reference or from the alternate allele. Not all of them, and I agree with you for those where you have a unique assignment to one or the other allele, it would make sense to include them. But for some of them, and we try to keep it as general as possible, you can't distinguish from what allele they are, or only based on the insert size distribution, and there's some uncertainty in there. So this is why we decided to keep them out for now. Maybe we understand it better in the future and decide to take them in again.
Thanks.
Alex, you had a question?
I have one question. Can you hear me?
Yes.
Okay. So, hi. Thanks for the talk. So I have more a philosophical question. I think in one of the first slides, you say that one of the limitations of the genotype pairs are how they combine different breakpoints for the same alleles. Okay. So my question here is, this problem must be solved by the tools that detect the variants or by the genotyper, in your opinion? So it's the tool like QTSV or PPSV that needs to be able to detect the correct breakpoints, or is the genotyper that must correct the breakpoints?
No,
and,
I mean, yeah.
What's your opinion? And the second question is, this problem is very common for insertion and deletions that are quite simple to detect from my experience. And I think that this problem will be even worse for complex structural variants. So do you think that it's a bit premature to move to complex structural variants when easy structural variants are still not that clean? So what's your opinion?
So the problem that we own for the single breakpoint versus entire variant is actually a genotyping problem. Of course, detecting variants, you often detect just a single breakpoint or novel junction, but then it's hard to report the variant. So most clean call sets of variants are report actually entire variants and not just single breakpoints. It's really a problem of the genotyping approaches that they only have a statistical model comparing the breakpoint spanning and novel junction spanning read pairs to each other, and it's one novel junction, one breakpoint. This is easily comparable. But when you have 3 novel junctions and 2 breakpoints, you somehow have to correct for this inequality in numbers of read pairs, and this is what people don't know how to do it in the genotyping process, not in the detection process. So it's really a matter of genotyping. And your second question,
it was...
is it premature? I don't think so. I think it's a generalization. So thinking about a problem as more general sometimes even helps to understand the problem better, is my opinion. At least for me, I understood the genotyping problem much better when working on this genotyper for a complex structure variant than when just thinking about deletion variants before, for example. So I think you can even find limitations of previous approaches when going more general. But yes, of course, you can always improve also simpler genotypers, right? And this is philosophy, I would say.
Okay, thanks.
Thank you. Maybe we can take a very last question because it's late already. Or no? So to my opinion, it's the first time I see a tool that works better on real data than on simulated ones, which is quite rare.
But only because we didn't simulate the inaccuracies at breakpoints. So we should probably also simulate that, and then we'll beat base type on simulated data, hopefully, as well.
Okay, so I think we can warmly thank you again for this talk. So,
thanks for having me.
Thank you very much. Have a very nice day, everyone, and see you soon.
Bye.
Bye.